Hi, everyone. Thank you all so much for coming to MIA today. We have two great speakers from UMass that'll be speaking with us. I won't say too much, just to let them have time to talk, but thank you guys so much for coming.
Thank you very much for having me. This is super cool. I had actually no idea this was a thing, and now I have a lot of YouTube to catch up on. So I'm sure my wife is going to be thrilled. But I'm going to give you kind of an overview more on what I've been working on really within the past year, working on the interpretation side of deep learning and what we can learn from these trained neural networks on regulatory genomic data. So my name is Greg. I am a bioinformatician in the lab of Zhiping Wang at UMass Chan Medical School in genomics and computational biology. My only disclosure is that in my free time, I even do more bioinformatics. So I definitely need more hobbies, I would say. Okay, so I think we all have seen something like this before. That's why we're here. But our work, we're particularly interested in the non-coding genome that constitutes 98.5% of our 3 billion base pairs. And in particular, we're interested in these regulatory elements, enhancers, promoters that regulate the 1%, and specifically in a cell type, tissue type specific manner, and in particular when this goes awry and its implications on human health and disease. So my work in particular, I study or I'm focused on transcription factors, and specifically about 1,600 transcription factors that bind DNA in a sequence-specific fashion. And for our purposes, I don't know why my computer's decided to not work, but as part of my work, I've curated, I think, over at this point, 30,000 ChIP-seq data sets or transcription factor ChIP-seq data sets from publicly available repositories. And we're up to about a total of 75% of these transcription factors have at least one TF ChIP-seq assay available across any of these 4 repositories. And specifically, but the coverage is uneven. So if we look, no surprise that we have about 3,000 data sets for CTCF. CTCF has pretty much been chipped at this point in every cell or tissue type you can imagine. But as I just took the top 60 transcription factors in terms of the number of data sets or publicly available data sets. And you can see that the coverage is quite sparse. So the focus of my work is, can we fill this matrix in? And if you actually zoom out on this entire matrix and you look across all 1,600 transcription factors and about 1,100 cell types that have at least one transcription factor ChIP-seq assay, it's about 1% sparse. So as much work as we have done in assaying these transcription factors in specific cell type contexts, there's a lot more information that we could learn or has still yet to be found. And then you can actually think that this matrix is actually infinite when you think about each cell or tissue has treatments, conditions, time points that could be assayed or studied. So, deep learning has transformed, I'm sure, given where I am, I'm sure deep learning has come up once or twice, but deep learning has kind of transformed the way, not only our everyday lives, but kind of the way we study regulatory genomics in the human genome. But for our purposes, we can think of a neural network as just taking DNA sequence and, learns features in the DNA, in the sequence itself, and transforms via neurons that sequence information into any form of output or prediction, whether it be a classification or a regression task. But unfortunately, the downside of neural networks is they're often thought of as black boxes. So we don't often, it's not easy to, or it's not often readily available or known what these neural networks learn in taking your input and making a prediction or arriving at a given output. And that's kind of the focus on my work. My focus on my work is kind of opening up this black box. So if you think, if you just train a simple convolution neural network, so convolution neural networks excel at learning features in images. Well, a DNA sequence, one hot encoded, is just an image in and of itself. And you just take a simple one kernel, single output CNN, and you train it on a CTCF ChIP-seq data set, you see that, and this occurs within a matter of seconds, you see that the convolution filter, if you just cast the values of the filter to nucleotides, you see that it begins to look like a CTCF motif. And then we can use some, very simple matrix operations very similar to what was done in some of the earliest of work in deep learning and regulatory genomics. So that being DeepBind in studying transcription factor binding, Bassett in studying chromatin accessibility. We can convert that convolution filter into a CTCF motif that everybody knows, recognizes, is familiar with. So we've leveraged this. We've used the fact that CNNs excel at motif discovery. We've shown that they outperform both classical and other deep learning approaches in motif discovery. And then we were lucky enough to partner with Eleanor Carlson here at the Broad as part of the Xenomia Consortium. So we're very thankful for her for inviting us, for being a part of that. And we used deep learning to essentially annotate transcription factor binding sites for 367 transcription factors. And then we're able to project all of these transcription factor binding sites into 2D space via UMass. And what we were actually able to learn is we were actually able to learn when specific transcription factor binding sites, so these specific regions of the genomes that transcription factors bind, we're actually able to see where they entered into the primate lineage. So that was really cool. So that was largely a focus of my dissertation work. And then we used that. We showed that not only do CNNs excel at learning motifs in TF ChIP-seq data, but we showed that that we can do this for any other kind of technology for assaying transcription factor binding. So showing here, we can do this for HC cell-X, so an in vitro high throughput method for assaying transcription factor binding. And then, as I am here as a representative of the Wang lab, our lab and the brilliant software engineers in our lab have built our factor book database. You can just go to factorbook.org and you can explore kind of all the data that we put together. Forgot that was 2 slides. And then we continued to kind of refine the way that we use neural networks to ascertain motifs in this case, a host of different technologies. And we continue to refine this method, and we were able to win the IBIS challenge, which was a kind of worldwide data science, think like a Kaggle-like competition, but specific to biology. And we're able to develop a method combining both CNNs and expectation maximization. So kind of old school bioinformatics method. It's crazy to say old school bioinformatics. Bioinformatics is a field that's only 20 years old itself, but that's how kind of quick this field is moving and how evergreen this field is. So that's really cool. I think that's just wrapping up. And then we've used this to, oh yeah, absolutely. Oh, yeah, absolutely. No, I have this one I'm wearing, right, I think.
Cool. That's all the problems.
Could you thanks for stopping me. I don't think I have enough slides to fill an entire, however long we have. So, yeah.
Can you comment a little bit on the experimental approaches to define transcription factor binding sites and what would you be I see some of them up there, but there were I guess recent papers about quote lower affinity transcription factor binding sites and contributions from I guess flanking kind of nucleotides and is it getting more complicated, our understanding of what's actually going on? Can you just comment on how you would actually define a transcription factor binding site experimentally?
Yeah, so for us, so if we're looking at, say, transcription factor ChIP-seq data, you take your reads, align them, you call peaks. Now you have your, essentially you have your enriched genomic regions that are bound by the transcription factor. But, if you've ever worked with, especially, ChIP-seq or, even DNase or attack peaks, right? They tend to be about 200, 300 base pairs long. But we know specifically with the transcription factor, they only bind a specific 6 to 20 base pair nucleotide sequence. And that's what I refer, when I say transcription factor binding site, that's specifically what I'm referring to, essentially the sequence within these peaks that the transcription factor is actually binding. And that's what I've used deep learning to essentially annotate these sites for any transcription factor that we have a data set for. But you're absolutely right in terms of, so if you were to say, there is some degree of correlation and it differs from factor to factor. But if you essentially take the peaks in a CTCF data set that have the highest signal, you'll find that those are your highest affinity sites. They're the ones where if you take the transcription factor binding site, that specific 19 base pair recognition sequence that CTCF actually recognizes, it has the best match or the strongest match to the canonical motif. Now, what you're in terms of now these, these low affinity sites, they're super important, right? Because, what's the prevailing theory? That these low affinity sites are the ones that can be easily tuned, or they can be the sites where maybe it's a CTCF site, but maybe it's also an SB1 site. So maybe it's a site where two TFs actually come together. And those are really difficult, right? They're really difficult for us to annotate. They're really difficult for us to define because what are they, right? We don't have like a curated motif for a CTCF SB1 hybrid. So it's a really challenging and ongoing problem. And I would say for me, and then the work that I'm going to discuss coming forward, it's difficult. That poses a problem because it's difficult to tell whether, you have this kind of, hybrid motif, but is it noise or is it truly a hybrid? So how would we properly annotate them, but then how would we also properly validate them? So it's a really hard ongoing problem, especially because these, as I was saying before, these hybrid motifs tend to be lower affinity sites. So they're the ones that, maybe they have a p-value slightly greater than 0.05. So when you call peaks, you don't even see them. So yeah, certainly, it's something that we're aware of and, we think about, but it's definitely hard to pinpoint. Yeah, great question.
There was a question on the chat.
How do I see that? It's pretty impressive. For a bioinformatician, I'm actually very computer dumb. So how do I see, or can someone read the question on this?
So the question is, can you detail how you transform the DNA sequences into images that can be ingested into a CNN?
Sure. So what we do is we actually, if you allow me to go back, actually this slide has it. Yeah. So what we can do is, so here's my kind of, toy nucleotide sequence I created. And actually in this case, I kind of inserted a GATA motif, right? So GATA is a transcription factor, so named because it recognizes GATA. So, but what you can see is what we do is we actually take this string and we actually cast it to an L/4 array. And when you stack them together, it becomes a tensor. But what we can do is numerically, we can convert the DNA string into essentially what becomes a matrix or array that is of length L by, in this case, 4, but you can imagine it's just by however many nucleotides you have or letters you have in your alphabet. So if you're looking at amino acids, it'd be L/20. If you're looking at DNA or RNA, it'd be L/4. So essentially, we just convert it to we call a one-hot encoded array of ones and zeros, where 1 is that nucleotide is present, 0 being that nucleotide is absent.
So you do 1D convention, basically just move the...
Exactly. And this is a great toy example. You essentially, so you have your convolution kernel here. This is what the DNA sequence actually, this is what the convolution kernel, that's what's actually learning. So it's, in this case, it's learning the motif, as training progresses. And that's what I'm showing. If I go one slide back, that's what we're showing here. We're essentially, here is your filter. These are your 1, 2, 3, 4 nucleotides. Here's your, what we would say is W is your motif width that you set in your neural network. Typically you set it to say like 32, something that's computer scientists, we love our powers of 2. It's, most computationally efficient. But for our purposes, we know that motifs are between 6 and 20 base pairs. So you can see that this convolution kernel, as the model learns, you can see the model learns it, the accuracy of the network, is approaching 90%. You can see that, that convolution filter that's randomly initiated, learns some biologically meaningful information. Yeah. Depends on, yeah, I got it. She was loud enough. It depends on what you're looking at. If we're training, in this case, this is trained on transcription factor ChIP-seq. So essentially anything that comes from a peak is assigned a label of 1. you can use, just randomly pull genomic regions and assign them 0 as long as they don't intersect a peak in this case. Or you can just, usually what we just do dinucleotide shuffling, so preserving dinucleotide content, and any other region is assigned to 0. Yeah.
So I'm wondering, for the ChIP-seq data used for the training of the model, so does the ChIP-seq consist of diverse tissues or very tissue-specific data sets?
So you can train in both. So if you say train on a single data set, we would call that single class learning or single class or single task classification. You can absolutely do that. You can do it in as you were saying, a multitask fashion, where essentially, okay, well, given all of the TF ChIP-seq data set, all of the transcription factor ChIP-seq experiments in a given biosample, cell, or tissue, you train on all of them at once. And that's actually, I'm not showing it here, that was actually part of my dissertation, was essentially looking at what happens when you train these neural networks in a single task versus multitask classification scheme. Because what happens is, sure, you can train a model to predict transcription factor binding for all transcription factors at once in a cell type. But what happens is that model learns cell type specific regulatory patterns. But those aren't then generalizable to an unseen cell type. The best example I can think of, and I'm sorry, I don't have the slides to show, but essentially take the transcription factor REST, right? REST is responsible for silencing neuronal genes and non-neuronal cell types. REST actually plays or is a cofactor with different transcription factors depending on the cell type. So if you take a REST data set that's trained in, say, HepG2, but you train on all of the TFs at once, it performs very poorly when you try to make predictions on an unseen cell type. So I guess the long story short is, yes, you can train on one label, you can train on many labels as you want, but you run into difficulty, and this isn't bioinformatics or computational biology specific, you run into difficulties when you try to make inferences in unseen cell types, and we call this essentially a lack of generalization. Cool, this is good because I definitely didn't have enough slides. All right. And like I said, my computer is just like, I don't want to work today. That's fine, me neither. Oh, this is working now. So we've used this as part of our factor book project to learn and annotate alternative binding sites. So this is essentially going back to the question of what we can see really well is when two transcription factors, they like to cooperate together within fixed proximity. So we see that really well. And the benefit of neural networks over classical motif discovery approaches is there's no need to essentially mask learned motifs. So if you've ever used like the meme suite to learn motifs in your data set, you essentially you learn a motif, you then mask those instances, and then you learn another motif. So you wouldn't see these homo and heterodimeric motifs because you would learn the monomer first, mask it, then you wouldn't see the two together. So that's kind of the advantage of deep learning, is we can learn these alternative hetero-homodimeric motifs because of there's no need to mask instances in the input sequences. And then, so sorry, give people a headache. So to kind of increase the complexity, so we can use very simple neural networks, CNNs, to learn motifs and transcription factor ChIP-seq, but we can, taking this kind of a step further, we can change, we're not going to change our input, we're still going to use DNA sequence as input. But what if instead we said, okay, can I learn the single base pair resolution ATAC-seq or DNA-seq signal? So our collaborators at Stanford, Anshul Kutaje, and his lab have developed ChromeBPNet, short for chromatin accessibility base pair resolution network. And what ChromeBPNet does is it actually consists of two neural networks. One neural network actually learns the intrinsic bias component of the assay, right? So what happens is you train a neural network first on non-peak regions to essentially learn the intrinsic enzyme bias, whether it be TN5 in the case of ATAC-seq or DNase1 in the case of DNA-seq. And then you use that, you freeze that model, and then you train a larger neural network to learn the individual bias-corrected chromatin accessibility profile. And all we're showing here is that here, it's, if you essentially take the number of predicted cut sites versus the number of actual cut sites in peaks, it does a pretty good job when looking, in this case, across chromosomes. So the neural network hasn't seen that data in that held out chromosome. What we can do is we can actually, so rather than just interpreting the filters as motifs, which is kind of the easiest way, we can run these networks in reverse. And when you run these networks in reverse, these attribution-based approaches for sequence interpretation, we can actually learn what individual base pairs in the DNA sequence contributed to making that prediction. So we do that, and I'm showing you here just kind of I actually don't even know what locus it is. It just looked nice. We take this, so this locus has a DNA-seq peak here. You can see that it's not this nice smooth peak we're accustomed to seeing from chromatin accessibility data, because all we're actually doing is we're just taking the 5' cut sites of the read. So we're actually DNase-1 made the cleavage or cleaved the DNA. And what you see is there's actually this bowl that's left behind. And then we can actually, if I chose a good color, you would actually see that we can accurately predict with ChromeBPNet the chromatin accessibility profile at that region. But most importantly is we can use these attribution-based methods to calculate nucleotide importance. And you'll see that the attributions sit perfectly right in the middle of that bowl, that divot that's left behind, because when a transcription factor binds DNA, TN5, DNase-1 can't get in there to cleave the DNA. And you zoom in on this locus, and you essentially cast these attribution scores to nucleotides, using the corresponding genomic sequence, and you see that they actually resemble transcription factor binding sites. And again, if I used a good color down here, you would actually see that the predicted chromatin accessibility profile has this really nice bowl, where otherwise in the observed signal, it's kind of more sparse. So what the network is doing is the network is able to essentially properly learn the underlying intrinsic enzyme bias, and then separate that and predict the true chromatin accessibility profile. And if you're interested in all this, they have a preprint in bioRxiv. It's really nice. I think I referenced it on the last slide. And then here you can see better because I chose a better color. You can actually see this works for a TAC-seq as well. And then same thing, you see the nice bowl where the transcription factor sits. And then same thing, you have the bowl that's left behind in the observed signal. We can accurately predict it, and those contributions or attribution scores fall right in the middle of that footprint where that transcription factor is indeed bound. So what we can do is we can actually scan these contribution tracks. So I kind of relate this to if you've ever used like a FEMO, find instances of motif occurrences. It's kind of part of our normal workflow for whether it be bulk ATAC-seq, DNA-seq, or single cell ATAC-seq. We essentially want to look for what sequence features are enriched in those peaks or those regions. So we always kind of scan, we use a library of motifs, and we scan the genomic sequence. Well, that's kind of the old school way. Well, now what if we essentially weighted the nucleotide sequence by how important that nucleotide sequence, how important each individual nucleotide is to making that prediction? Well, now we can kind of scan genomic sequences, but in a cell type specific context. And what we see is when this happens, the peaks, whether they be DNase or ATAC-seq peaks, we observe this kind of bimodality where you have your set of open chromatin regions that do not possess a transcription factor binding site for a given TF, and those that do. And what this looks like in the real world is it looks like this. So what happens is we have these transcription factors exhibit this bimodality in the cell types in which they are active or expressed. So kind of cherry-picked 3 TFs here, CTCF, SP1, and NRF1, showing you here the gene expression, just pulled the gene expression from ENCODE. You can see that those 3 TFs are about evenly expressed across these 3 cells. We're looking at HepG2, so a liver cell line, GM12878, a white blood cell line, and then K562, a red blood cell line. And then you see for CTCF, you see that indeed CTCF, we see this bimodal distribution. And then what we're also doing here is we can actually use this bimodal distribution to derive a threshold for essentially whether or not we see an instance of that motif or not. And if you've ever used like a FEMO to scan your genomic sequences before, there's always this problem of what p-value threshold do I set? you tend to find that different p-values are better for different length motifs, different motifs of information content. This kind of takes care of that problem for us. And then what you see is if you take all of the transcription factor binding sites, so the individual 6 to 20 base pair nucleotides corresponding to the underlying transcription factor, and you aggregate both the observed and the bias-corrected signal. In this case, I think it's DNA. So you see that indeed you have a bowl for all of these transcription factors. So not only can we pluck them out, we can observe their footprinting. So you can also think about it as another means of a footprinting method. But like I said, we see this in we see this in a cell type specific context, just like my clicker likes to work in specific contexts. So we see that in HepG2, HNF4A only exhibits this bimodality. HNF4A, I'm sorry, only exhibits this bimodality in HepG2 or hepatic or liver cells, where otherwise it does not. Same thing for NF-kappa B. We see that it only exhibits this bimodal pattern in white blood cells, but not in the liver cells or the red blood cells. And then lastly, you can see where we're going with this. Again, HNF4A only expressed in the liver, NF-kappa B1 expressed only in the white blood cells, and then GATA1 only expressed in the red blood cells, and it's only bimodal in those red blood cells. So just kind of going in a little further, we can look at CTCF here. So what we actually have is we actually have 1,500 of these trained models corresponding calculated attribution scores for 1,500 combined DNA syntax-seq data set from the ENCODE consortium. And this work is, essentially is part of kind of phase 4 of the ENCODE project. And if we essentially just take the difference in the two means of the populations, just using a 2-component Gaussian mixture model, some questions I always get is, 2-component Gaussian mixture model doesn't seem to fit great. This actually ties into the question before about, essentially these composite motifs that resemble both, more than one transcription factor. That tends to be what this middle regime is. So if you actually convert this into a 3-component Gaussian mixture model, you'd see one here and then one there. Essentially, you would see that kind of that murky population. But for our purposes and moving forward, 2 works fine. But you essentially see that if you calculate the difference in the two means of the two populations, you see that CTCF, a ubiquitously expressed transcription factor, that difference in those two means is always great, right? We expect CTCF is active in every cell line, master regulator, Swiss Army knife of the human genome, to be corny. But if you look at HNF4A, again, our liver-specific or hepatic-specific transcription factor, we see this bimodality. So if you take the difference of the two means, we use that as like a surrogate for bimodality, right? Obviously, we do statistical tests as well, but we can see that it's HNF4A only exhibits this bimodality in liver and related tissues, so intestine, large intestine, liver, etc. Well, to this point up to this point, we've only presumed that the given transcription factor is bound at these TFBSs that we've plucked out from these open chromatin regions. But if you then essentially you go back and you intersect these TFBSs against all of the ChIP-seq data sets that we've curated, you actually find that even in the best instance, say CTCF and REST are bound about 80% of the time to these sites that we've plucked out. And this is mostly also due to the fact that these transcription factor binding sites don't often perfectly match. They look like one, but it kind of a threat could still be a thresholding issue. And then this is just to highlight the fact that we understand the kind of the limitations of this work is the fact that you can really only do this at a transcription factor family level, right? We know that CTCF and CTCFL have, for the most part, indistinguishable transcription factor motifs. Now we can use the fact that CTCF is expressed everywhere. CTCFL is only expressed in the germline. So we can use other lines or orthogonal lines of evidence to essentially we know that most likely it's this transcription factor member of this family and not the other. So essentially just showing you here that as part of this work, we've essentially taken 620 TF motifs derived from ChIP-seq data, and we've clustered them into it's about 350 unique clusters based on their binding specificity. But here, just highlighting again, it's more likely if that transcription factor exhibits this bimodality, it's more than likely going to be bound. But can we make a more accurate prediction about whether or not that transcription factor binds this TFBS that we've plucked out. Well, what we can do is we can take all of these, Chrome BPNet associated tracks. So we can take our observed signal profile, our predicted signal profile, the bias corrected profile, and then these contribution scores, as well as evolutionary conservation, because why not? It's just another, tacking on another vector. And we can use that one hot encoded DNA sequence at these essentially putative TFBSs or transcription factor binding sites. We can pass them through just a very small neural network, and we can actually make inferences or accurately predict that a transcription factor is bound at that location or TFBS that we've plucked out. And more specifically, if a transcription factor exhibits this bimodality in both the training and the testing cell type, we can accurately make inferences across cell type. And this goes back to, we talked about in a previous question, we talked about, the lack of generalization or neural networks. One of their downfalls is their inability to generalize across cell type. And that's really the focus of my work, is to be able to make accurate predictions across cell type and know specifically when we are confident in making those predictions. So highlighting here that essentially, okay, well, you take DNase in K562 and you train, and then we're also training cross cell type and cross chromosome. So if you've ever done this work before, I train on all chromosomes but chromosome 1. I leave chromosome 1 out. And then when I make my inferences across cell type, but also cross chromosome. So if there are any sort of patterns that are specific to a chromosome with respect to transcription factor binding, the model will never see it. So we can accurately make inferences, within DNase in K562, cross cell type, cross chromosome. We can do that. We can also, we can do the same thing for attack. Given attack-seq in K562, we can accurately predict transcription factor binding in HepG2. And then the coolest part is, again, really cool that my clicker decided to not work right there. We can actually, if you have a model that's trained on DNase, you can actually use that model to make inferences not only cross cell type, cross chromosome, but also cross assay if you have attack-seq data. So this is actually really cool because the ENCODE consortium has dozens, if not hundreds, of very deeply sequenced DNA-seq data. But for the most part, due to costs, the number of cells required, at this point, most people just do attack-seq. But we can leverage all of that information that we have as part of the ENCODE consortium to make inferences cross cell type, cross assay in essentially unseen attack-seq data. Now, what can we do with this? And so what we can do is, well, okay, so now we know which TFs we can accurately annotate and predict they are bound in a given cell type. We can construct this matrix of, say, we can take cis-regulatory elements from ENCODE, so promoters, enhancers, insulators, silencers from the ENCODE consortium, and we can annotate how many times, this is just a toy example ChatGPT gave me on the train today, and you can essentially say, okay, well, we can calculate the number of times that a given cis-regulatory element harbors a transcription factor of interest, again, the ones that we know we can accurately pluck out. And then what we can do is we can embed this matrix of integers, again, into 2D space using UMapp. And you actually see that in this case, about 100,000 cis-regulatory elements that are open active in HepG2, they actually form many distinct clusters based on cis-regulatory syntax. So essentially, which transcription factors they harbor transcription factor binding sites for. And here I'm just highlighting, again, highlighting that if you, just plucking out this specific cluster 3 here, that this cluster 3 are your HepG2 specific cis-regulatory elements, open chromatin regions, again, promoters, and these are enhancers in this case. They're all enhancer-like, they're all distal from a TSS, but they're all the ones that harbor transcription factor binding sites for HNF1A and HNF1B. So we can use this to begin to essentially, interrogate these cis-regulatory elements in terms of, not only are they active, but why are they active? Who's contributing to their activity in the sense of who's binding there? So then lastly, what we can do is, well, we just looked at one cell line and we said, okay, well, in HepG2, we're able to annotate which transcription factors are binding to which cis-regulatory elements. But if you look at one cis-regulatory element across a host of cells and tissue types, and here I'm just essentially showing you across, I think this is actually, it's combined bulk attack-seq data with also single cell single cell brain data up here, you actually see that you see different transcription factor usage across different cell lines. And we refer to these regions as polysyntactic. So they're open across several cells and tissue types, but they do so, or they're leveraging or using different transcription factors and different transcription factor binding sites in their activity. So this is kind of part of my work is, how often, obviously I can just generate a bunch of PNGs and I can just pick out which one looks best. But essentially, part of our work is essentially, can we calculate or can we quantify how often cis-regulatory elements exhibit this polysyntactic behavior? So that's kind of like kind of an offshoot of this project that I'm working on. And I think that's it. Yeah. So, I just like to say, thank you, very much for having me. This is, a really great opportunity to highlight all of the great work of the Weng Lab and Moore Lab. We're essentially one giant conglomerate or entity at UMass Chan Medical School. But we'd just like to thank, all of the lab members, all of our HPC admins that, anytime I break something, they fix it for me, which is quite often. So yeah, and with that, I would be happy to take any and all questions.
Thank you. Great presentation. For your last slide, where you showed basically like in different cell types or cell lines, these different regions can be active due to different transcription factors. How much does that correlate with like the expression of that transcription factor in those cell types? Like, would you be able to just infer that based on like what transcription factors are active?
Exactly. And it comes down to, at the end of the day, those transcription factors do end up being the ones that are expressed. But we just don't know how often this is occurring. We don't know, I know for a fact this is like an enhancer region. Is this enhancer regulating the same gene in one cell type as it is another? So questions like that as well. But yeah, absolutely. You can trace this back to the gene expression, just like I did like a couple slides ago.
Thank you.
Hi.
Great. I wanted to ask about if you can go back to the bimodality, like in the next example, I guess HNF4 was it that you have more zoomed in? This one. So yep. Here, like I wouldn't have said that this is bimodal. So no matter what, you try always to fit a bimodal distribution. Like how do you evaluate after, like for something that is less known? You say, okay, let's assume that this is bimodal. You fit the distribution and then you see some scores. Do you rely on literature information? How do you know that is not like just because you're going to always divide something, you're going to get something?
Yeah, exactly. And actually in talking with, Jacob's been a huge help on this project too. And that's exactly something, the first thing he brought up was, how do you know you're just not artificially, fitting two means where you should really only have one? Right. And so right now we just use the difference in the two means as our surrogate for bimodality. Now, I've also, incorporated more statistically rigorous methods like Hartigan's dip test to, essentially assign a p-value for how much the distribution deviates from, unimodality, I guess, for lack of a better term. But that actually, wouldn't actually catch this case. So it's definitely, it's a technical, certainly a technical problem that we have to push through, because what happens is you actually see, you can imagine a mode there, like you could fit as many Gaussians as you wanted to this, right? So essentially you see like there's like a Gaussian here, and what happens is it actually turns out to, these are the sequences that perfectly match the CAAG in the motif. But then you see the population here is the ones that only slightly deviate from the consensus. And the problem is because, motifs are a different length, they don't just, it's not a picture perfect bimodal unimodal pattern. So yeah, it's definitely an ongoing technical problem that we have to solve because of the biological implications.
And kind of related to that, and I remember you showed me this a long time ago, but for how you presented, you rely a lot, of course, on the motif. Like ChromaPinet usually uses Modisco after, like, you know, because the sequence attribution score per se can still be, I don't know what I'm looking at, right? So you either, so in your case, you're relying on the motif. But I remember you in some instances, you improve the motif, right? Like you are able, so I guess what I'm asking is how good does a motif need to be for you to catch it? Basically, how robust it is, and also through this method, can you actually improve Jasper database? And
so that's part of this as well. So essentially, what I've done is for this, every TF that went into this heat map is a TF motif that I have hand curated. So, and again, it's kind of, you know, unfortunately circular in the fact that I'm the one who curates the database and then I'm the one doing, so like, you know, it is a bit circular in that sense, but I'm essentially hand curating using the most information-rich motifs to do this essentially scanning process. Because, you know, you sound familiar with TF Modisco and using secret-based methods to do de novo motif discovery from contribution scores. And sometimes those motifs don't, if you say like run TomTom against those discovered motifs against the literature, aren't fantastic matches, right? So that's why I use the hand curated ones from ChIP-seq, you know, using like this, just the very simple kernel scanning method, which I find gives better motifs than the attribution-based methods.
And so how many are you left with from 1,500?
So 620.
620? 620.
So they're the only 620 that I am confident in from the ChIP-seq.
Yeah, no, that's fine.
Yeah.
I have another question. Nobody else has it though. This is for the future.
Yeah.
I mean, there's been, I think someone in the lab did it, but there are other papers that showed that, of course, a lot of these motifs are more sensitive to others, to genotype variation. They show allele frequency, allele specific binding. So have you thought, it's in your future interest to try to incorporate genetics and try to characterize them more in like go to the, basically to the clinical, like look at variations and try to annotate which one are more which one are more likely to respond to a SNP variation, more causal to or something, and intersecting with GWAS, this kind of things.
Exactly, and that's like that'll be essentially hopefully you know, tying this all together will be exactly that, intersecting all of these. Essentially, we'll use the 1,500 ENCODE experiments, DNA syntactic experiments, to essentially call an atlas of these transcription factor binding sites, and then that'll be one of the first things we do, is essentially intersecting them with all the GWAS and all these SNPs to do exactly that. Right.
So this is gonna be just posterior, like
exactly, yeah. So in the case that you mentioned, no, that's not, hasn't been using, like looking specifically at allele specific binding. No, that's not in the immediate future.
I think someone did in what you call the TFs, right? Or at least what was available at the time. And did an allele specific study on that.
Yeah. Unfortunately, that's still sitting on.
But this is the published.
Oh, published.
I think someone did it.
Okay.
I double check.
Yeah. Show me. Yeah, show me.
So in your last slide, again, bringing in the genetics here, I don't know if you mentioned, for example, that you don't necessarily know if this different elements through binding to different TFs will even regulate the same gene in different tissues, etc. So I was wondering if you have already a few examples, or if you plan to have some in the future of specific genes and how that's, or maybe take these regulatory elements that we know what gene they regulate, at least in some tissues, because, for example, it's been proposed that more essential gene, for example, they have more complex regulatory landscapes. So that might actually be very interesting for someone to see that the same gene has different, is being regulated by a different enhancer or a different pattern in the same enhancer in different cell types. So I think that would be very interesting. Starting from genes that we know the enhancer gene link.
Yeah. Yeah, exactly. And we hope to, that's what part of what we hope to do, especially leveraging all of the 3D chromatin interaction data we have from ENCODE is absolutely do that. Yeah. My to-do list just keeps getting longer and longer, as I'm sure all of ours does.
One more question, if we, yeah, I think you briefly touched upon that. I wanted to ask if there is anything more you can comment on, like the cooperative or synergistic effects of the different TFs, or if you have any way of pursuing this further. Yeah, like they both have, or yeah, let's say pairs for now.
Yeah,
they both have to bind to, like, how does that factor in how much more complicated would it be to systematically
so it's easy, no, I don't mean but it's easier. Sorry, as I give everyone a headache here, I don't remember where. Do I have it here? Yeah, I do. So it's easier when you're looking at essentially two TFs that we know bind at a distance. So we know that FOS and T cooperate together. And we actually know specifically they cooperate together in a lot of cancerous cell types. And we actually know that actually not only do they prefer, but we actually know that they prefer to be separated specifically by 6 base pairs. And you can do the analysis. You can actually, you can take these two motifs and you can slide them across one another, and you can see the changes in the predictions of the binding or chromatin accessibility. You see it actually, spikes at that 6 base pairs. So when it's two motifs that, are separated by a specific fixed distance, we can see that really well. When it's more murky and it's essentially the two sites overlap one another, and you kind of squint and it looks like one motif, and you then squint and look the other way and it looks like the other, that's very difficult, and that's something that's very hard.
Do you have any sense as to how often these two motifs appear together versus separately in the whole genome? Or like, is this a trend that you see in the entire genome, or I don't know, very local?
Yeah, so in this, I can tell you specifically in this specific instance of the essentially FOS-T dimer, there are thousands of these. And specifically, they're enriched if you were to run motif discovery on like MCF7 cells, A549 cells, a lot of these cancerous cell lines, you'll see an enrichment for these, and it occurs in the thousands. Now, in terms of like when one versus when they occur exactly at 6 base pairs versus not, I don't have that exact number. But that specific, this specific instance is, I don't know if it's in the literature that it prefers to be 6 base pairs. I know that it's that they prefer they're always together. But yeah, that analysis is, or that like field is still in its infancy.
Thank you.
Yeah.
Well, thank you so much, Greg, for speaking. That was a great talk. I think we'll do like a 5 to 10 minute break before we begin the next talk. We're now going to be moving on to the next talk of programming for MIA today. So we'll be hearing from Jacob Schreiber, who actually just began his lab at UMass Chan in Worcester. And we're very excited to hear about his work.
Thank you. As Laura mentioned, I just started my group at UMass Chan Medical School, and so it's kind of fortunate timing that I get to come here and introduce to you a little bit about what I've been doing over the course of the last few years. But before we talk about that, let's take a little bit of a step back. Over 20 years ago now, the first draft of a human reference genome came out, and a little snippet of it looks something like this. In the words of a familiar face, Dr. Eric Lander, genome, bought the book, hard to read. Even though it is challenging for us mere mortals to understand these long sequences of A's, C's, G's, and T's, having this map provided the basis for the next generation of biochemical assays to be developed. Here we can see 4 such experiments, each measuring the binding of an individual transcription factor. Just by looking at these readouts, we can see how informative these sorts of experiments are. At this enhancer, there's the binding of OCT4, SOX2, and NANOG, but not necessarily the binding of KLF. However, relying solely on the readouts from these experiments is kind of reminiscent of the blind men trying to describe an elephant based on what they felt. Just because we understand what is happening and where in the genome doesn't mean that we understand why it is happening there. And so enter sequence-based machine learning models. Greg gave a great introduction to the sorts of models that I'm talking about here, but at a high level, there are many different internal computational units that these sorts of models can have, convolutions, transformers, et cetera. And there are many different precise formulations of the tasks that the models try to solve. However, broadly speaking, sequence-based machine learning models are those that take in nucleotide sequence and make predictions for the outputs from these sorts of biochemical assays. But the predictions from these models, in my view, are kind of some of the least interesting aspects of them. Most of the assays that we deal with provide us with genome-wide readouts. There is no more like secret genome that we need to go and make predictions for. Rather, in my view, one of the strengths of these machine learning models is being able to run them backwards, to be able to get them to explain why they made the predictions that they did. And if we look at this example here, we can see that the reason why this model predicted the binding of OCT4 here is because there's an OCT-SOX heterodimeric binding site there. So over the course of the last year, I've been working on basically trying to take this mechanism and repurpose it for design, but specifically the design of edits to biological sequences edits to biological sequences. The way that this works is basically as follows. Just like before, you have some sequence that you're working with, you have your machine learning model, but now you have a new component, the desired output from the model. Maybe you want to design a region where proteins really bind very strongly. Maybe you want to design a region where the protein doesn't bind at all, or one that's a site that's accessible, a site that's transcribed, et cetera. Anything that can be the target of machine learning prediction. The way that this works is that you take your sequence, you run it through the model, and you get what the actual output from the model is. Now you take the actual output, you compare it with the desired output, and now you run this difference backwards through the model. Rather than being able to highlight the nucleotides that are driving model predictions like we did before, we are now highlighting the nucleotides that need to change in order to achieve your target objective. But we encounter a problem here. This design of edits problem is discrete. We have to choose which positions we want to edit and what characters we want at that position. But the values that we saw before were quite continuous, where the height of the character basically corresponded to how important it was to the model. So the main technical challenge that we had to overcome with our method was recasting this discrete optimization problem as a continuous optimization problem that can be solved quite simply. So the way that we did this was we introduced this method called Ledidi that ends up learning an underlying continuous weight matrix. Let's take a look at what's happening midway through this design process. We have our initial sequence that we're trying to edit. At this point in the design process, Ledidi doesn't really care what it is, maybe has a slight preference for C. And what we end up doing is sampling the original character, the G, at that position. Then at the next position, it's originally an A, but Ledidi thinks that quite strongly it should be a T here. And so we end up drawing a T, an edit to that sequence. We can go down the entire sequence, mostly keeping with the original sequence, but at some points making edits to the sequence, and we end up with a new edited discrete sequence. We can run that through the model, get our actual output, compare it to the desired output, run the difference backwards to the model as before. But when we encounter the edited sequence, we can just go straight through it and end up optimizing this underlying continuous weight matrix. In this manner, we have recasted the discrete optimization problem of choosing which edits to make as a continuous optimization problem that can be solved using just off-the-shelf optimizers and really just a few lines of code. What's a little bit more complicated, though, is the exact loss that Ledidi is optimizing. It's comprised of two terms. The first is the output loss term that most design methods have. Are you achieving your desired objective? Does this sequence make the predictions that you would like it to? The second is this novel input loss term. It's basically just counting the number of edits that were made in that sequence. And so in this manner, we are explicitly trying to minimize the number of edits necessary in order to achieve our target objective. Then, of course, because there are two terms, you have to have a mixture weight there that controls the balance between the two. And the rough intuition here is basically you set it to however many units of the output loss you want each edit to be able to reduce by. So it takes a little bit of tweaking, but it's actually usually quite robust to this. Then the first question is, does this sort of formulation work? And so we paired it with a situation I initially thought was going to be quite simple, but ended up being quite interesting afterward. This was the design of GATA2 binding sites. Greg introduced in the earlier talk that GATA is quite a simple motif. It binds basically to the motif GATAA, and that's where I expected it to land. And so we took some sites that initially were not predicted by a BPNet model to have GATA binding, the black line there, and we wanted them to be edited to become a site that GATA bound quite strongly. And after running our process, we end up with sequences over there. They don't quite end up on the cyan line because remember, we have a mixture of two terms, and that if we got closer to the cyan line, that would cost too many edits to do. But basically, we're still able to design sequences that had strong GATA binding. Yeah. Oh, sorry, I think we need the microphone for the people online.
About the loss term, because then you say the number of edits, but that's the discrete thing. So it's like you're sampling, so it's like you're penalizing the probability difference. Like, can you just talk a bit about what that?
It's quite literally the discrete number of edits, and so it ends up being an integer loss, and you can work this into a differentiable loss in the same way that you'd work an L1 loss in the sense that it's. We can talk a little bit more about it afterwards, but it's not that complicated.
Okay. Yeah. So can you say a little bit more about the Ledidi matrix itself? I was going to ask, it's if every row, does it sum to one? Like, is it a probability of finding each nucleotide?
Yes, that's a great question.
Like, is it parameterized somehow? How its dimension?
It's a really good question. Sometimes I skip over the technical aspects here because depending on the audience, they don't care. But it's nice to encounter an audience that does care. The gist is basically that this weight matrix is initialized to all zeros, though you could potentially initialize it to very small random values, depending on if you want to run this multiple times and get something different. It's initialized to all zeros. And what you end up doing is you interpret the values of these weights as logits. And so it's not that you sample from the Ledidi weight matrix, it's that you sample from the addition of the initial sequence and the Ledidi weight matrix. And so what you end up doing is slightly preferencing that probability distribution towards the initial sequence, but still allowing the weight matrix to say absolutely not, if it thinks so strongly. Does that make sense? That's the reason there's the plus sign there, that you are sampling from the addition of the initial sequence and this weight matrix. But another little detail here is that it's not just the one hard-encoded initial sequence, it's basically the log of the initial sequence plus a small epsilon term, where when the value is like a G there, that's a 0. And for the non-G positions, it's like -4 or so. In principle, that's another hyperparameter, but usually I found that it doesn't really make that much of a difference. That it just controls the dynamic range of the final values.
Okay. Thanks.
Thanks. On the next slide, could you just re-explain, like, what's the starting sequence you use before you start making these edits to get the GATA2? Like, is it a random sequence, or and does it depend where you start in terms of what you get?
That's a great question. We're going to hopefully talk about that exact second question about differences in how you start a little bit later in the talk. This is entirely a toy example meant to just show that Ledidi works in principle. So here we took regions that were not GATA peaks. I didn't do anything particularly clever here. I just took real genomic regions that GATA did not bind to, and I set it to a very high value, and I wanted to see whether or not Ledidi could create edits that caused you to have GATA binding.
This is such a biologist question. I'm sorry. I feel a bit like a fish out of water in this seminar. But how big is the region? We think about transcription factor binding a lot. And how big is the region compared to like nucleosome binding?
Yeah. I'm going to remake this point a little bit later, which is that Ledidi is a design algorithm, which means that it can be paired with any machine learning model. And so the receptive field you are doing the edits on depends on the model that you're using. Here, this is a BPNet model whose input window is intuitively 2,114 base pairs. And so that's the size of the edits, the window being edited. Cool. Okay. So if we look at the losses, another question?
So how do you actually optimize the loss here? Do you use gradient descent or some sort of like sampling, like genetic algorithm or something like that?
Yeah, that's a great question. I just use the Atom optimizer. I use Atom because everybody uses Atom. And specifically, I use the Atom implementation, which has a bug in it, and not the Atom W implementation, which has fixed the bug because everybody uses the Atom implementation. Or maybe not bug, but it misses a technical detail that from the original, but that's what I use. You can use any optimizer that you would like. Really just, if you're using PyTorch, which you should, it's the optimizer that you use is completely decoupled from the design process.
Yeah, thanks.
Okay. So if you look at the losses during the design process, you see that there are basically 3 regimes. In this first one, the input loss rapidly increases. Basically, edits are being proposed rapidly in order to try to meet our desired objective. In the second regime, these obviously unhelpful edits, kind of like edits which are in LD, if you think about it from a computational perspective, with the ones that are actually valuable, end up being discarded because we're trying to explicitly minimize the number of edits that are being proposed. And then there's this third long regime, which is a long tail. Basically, that we are slowly merging sets of slightly helpful edits into smaller sets of moderately helpful edits, basically trying to see whether or not we can condense the size even more. And if you look very carefully there, you can see that roughly you're reducing the size and number of edits by 33% over the course of, that 150 steps there. So what edits are actually being proposed? If we look at the GATA site, you might be unsurprised to see that adding in GATA binding takes 5 nucleotides when the GATA motif is GATAA. But if you look at the exact positioning of where these edits are happening, you see something really interesting. Only one edit, that really big A, is happening in the core GATAA site. 3 edits are being made in the surrounding flanking regions, which is another point that Greg alluded to in his talk, which is that the flanking context of these motifs can matter a great deal. 1 edit is kind of out far there. You may initially want to discount what's going on there, but what it's doing is it's adding in a tau binding site. Tau is a cofactor of GATA, which greatly enhances its binding. And so what's happening here is that not only are we making the GATA site nice and easy for GATA to bind to, we are adding in a cofactor site. And so Ledidi is specifically targeting a site which was almost GATA, almost a strong GATA site, and specifically making the nucleotide changes there, as opposed to just plop it in a motif somewhere random in the sequence. If we use a simple pruning algorithm, we can actually get this down to 2 edits and still preserve 98% of the predicted binding. So maybe you're still not impressed. Maybe you think GATA is quite a small motif. It's still not surprising that 5 edits adds in GATA. This was quite pervasive. If we look at CTCF, you can see that with just 4 edits or 3 edits after pruning, we were able to go from no CTCF binding to strong predicted CTCF binding by targeting a site in the genome that was almost CTCF. I have a kind of evolutionary hypothesis about this, which as a computational person, I'm not particularly well equipped to handle or even super formalize, which is basically goes like this, that our genomes are a graveyard of past viral infections, transposon activity, all sorts of other things that used to do stuff which was bad. And our genome evolved out that function in part by disrupting key binding sites. And so we can use Ledidi to bring back these binding sites in ways that you might not intuitively expect. So I think about the genome as being much more kind of continuous than you might normally expect, that there are so many sites that are almost motifs, much more than you would normally expect. Just for time reasons, to make sure I get to everything, I'm going to skip this. An important point here is that because Ledidi is gradient-based, it involves running these values backwards, it ends up being quite faster than other approaches that are out there. If we try a simple approach where we take around 17 motifs that are relevant for the design task that we care about, and we try plopping them in at every position in the sequence and do our design that way, in this method which is called motif implantation, it takes a few hundred seconds to do the design task using this model called SAY. If we try greedy substitution, which is sometimes it's called directed evolution, where you make one nucleotide edit at a time until you achieve your desired objective. It takes around 200 seconds to achieve your target goal. If you use Ledidi, it can be over an order of magnitude faster. And the reason for this is because you can imagine that greedy substitution is basically just going through and saying, does this edit help? Does this edit help? Does this edit help? For every possible edit that one could make, kind of like a petulant child. In contrast, Ledidi potentially can make multiple edits at the same time. By using the gradient, it can even add in entire motifs. And so each step, even though it takes slightly longer because you have to go forward and backwards as opposed to just forward, adds in more information to the design task. You take bigger steps towards your goal. I'm going to skip this also for reasons. So, like the point I was making before, Ledidi is a design method. Sometimes when people publish papers talking about how they have a new design method, what they really mean is they have a really cool new machine learning model that they have then applied standard design methods to. And those machine learning models are cool, and the ultimate designs they get out of them are cool, but they haven't actually proposed a new design method. Because it is a design method, Ledidi can be paired with any model that's out there. Any model you've already trained in your lab or have from a collaborator, any model that's already public and out there. And so in order to demonstrate this, we tried to apply Ledidi broadly, just using out-of-the-box settings. Here's a different version of our GATA friend from before, where we take a region that exhibited median activity in the genome, so not particularly high, not an anomalously low region, but a region where GATA didn't bind. And we wanted to turn it into a really strong region where GATA does bind. And we're able to achieve that, like we showed before. But then we considered 4 different transcription factors across 2 different models. Out of the box, we were able to design edits that achieved our target goal quite simply. Then we looked at 4 different models of chromatin accessibility. And again, saw that we were able to achieve our desired goal without changing the settings at all. Then we looked at transcription initiation. We looked at MPRA activity. We looked at STARR-seq data. And in each one of these settings, Ledidi was simply able to design edits using the model in order to achieve our target goal. So Ledidi is a general purpose designer of edits. All right. Sometimes this term overfitting comes in when people talk about training machine learning models. They're worried that the model has overfit to the training set and is not actually generalizable in practice. Here, it's important to keep in mind that the same concepts can apply to design, that potentially the designs that you create can overfit to the parameters of the model. Maybe the model has learned something wacky which doesn't actually hold. Maybe you're able to push the model out of distribution in some wacky way. If you think about computer vision, there are a series of papers that came out that showed that if you had an image, you could change 1 pixel value in an imperceptible way to get it to predict any class that you wanted. It's not quite as easy to do with DNA models because you just have discrete characters, but that sort of idea is potentially valid. And so a question that we had is, are the edits that we're proposing actually generalizable, or do they, or are they just pushing the model off distribution? And so what we did is we took edits that were designed using BPNet models. Each group here is for a different transcription factor, and each color is a different external model. And we wanted to verify, if we design edits using BPNet, do external models also predict higher, bind? We were considering mostly just higher binding here. Do these external models also give us higher binding? And even more indirectly, if we consider models of chromatin accessibility, if we design edits that say add in CTCF binding, do models of chromatin accessibility predict higher accessibility? And so in each one of these cases, we saw that we were able to, that the edits that we designed were generalizable across models, and we were able to validate. Yeah.
If all the models predict that pixel, does it make the pixel biologically relevant? Like, just because they agree that that pixel is important doesn't make the pixel relevant. Like you said, you can still predict every class. So they just agree on.
Yeah, so here the idea is that we're trying to not overfit to the eccentricities of any specific model. It's unlikely that very different models trained on in very different ways on different processed forms of the readouts, even different forms of biochemical assays, would have the same eccentricities.
Sure.
Like I think in the case where you take a pixel and you are able to modify it slightly to get any prediction that you want, that if you then fed that into a second model, you would not get the same response. That it's just an eccentricity of the specific model you're working with.
I see. And when you write Ledidi plus pruning, is that the pruning is post or is something that just happens?
Oh yeah, that's an optional post-processing step afterward.
Like the you do, or
there's built-in functionality.
It's a built-in
function. What we have is, I mean, you can do whatever sort of pruning you want, but we do a greedy approach where you go through every edit and you see what happens when you revert it. And if reverting it doesn't bring you, there's a tolerance for how far away from your desired objective you can go during the pruning thing. And if the edits are within that tolerance, then you undo them.
Because I was just seeing that you go to just a couple of bases, I would be curious to see from BPNet, for example, attribution scores, like if you're picking the top 2 most important from BPNet, like in a certain kind of way, and trying to understand the like how you use it and like.
So are you saying for the pruning step?
Yeah, in general, because you cannot make that many edits, like you showed on the first output. Mm-hmm. And so, but in the end, after you prune, you get just 2 bases. Yes. And maybe it was just a case, but like if those 2 bases are the one that from BPNet gives a very low attribution score, meaning that if they're not there, there's no accessibility. If you pick just the top 2, the model will tell you that now there is accessibility, but maybe biologically will not work. Like.
I think this is an interesting conversation that we should talk about later. I think that ultimately, you're right that there are many sorts of post-processing strategies that one could use. I generally try to avoid getting into this because there are so many heuristics that one could consider. I was more just trying to make the point that we were so close to a GATA site and you might not have known it if you had only looked at attributions or if you had only looked at predictions from the model.
Are you going to talk more about experimental validation, or is
yes?
Okay.
Yeah, quick question about the baseline. So, what do you mean by same edit composite? Because I'm trying to understand whether this is a result about the consistency of these models or about your approach to find something that is consistent.
So the same edit baseline is that if we take the same number of edits of each type, like A to T transitions, etc., and we apply them randomly 100 times in that sequence, what do the model predictions look like?
Okay, but there's different ways to think about what same is going to be in that sense. So for example, as you're saying, it's like the frequency of one to other, but then you've also got, for example, like, do they match up with the attribution, the bases that are contributing to the out, like do you include that information? So it's like,
remember that these are sequences that do not exhibit the activity normally. If you look at the attributions, there's going to be nothing because the models are not predicting that there's binding. So you can't really use the attributions in order to guide where you want to make edits.
Right. Okay, I see what you're saying. But I mean, you could take the gradient or something in this particular direction, like try and control for.
I mean, if you start to talk about you taking the gradient in a certain direction, then you come up with Ledidi, right? Because that's what Ledidi does. It uses the gradient.
Okay, but then just in general, do you think this is saying something about Ledidi or about the consistency of what's driving these models?
I mean, I think ultimately it's both, right? That if one model fails to pick up on a motif that actually is biologically plausible and a second model is picked up on, like you're not going to see as strong like there's a reason here that CTCF gives us the cleanest signal, right? In terms of if you design a CTCF binding site, that Informer really agrees that that's a CTCF site because CTCF is so simple. These other ones have more complicated binding modes. And so there may be more disagreement about what exactly is the strongest version of each motif amongst them. So, I mean, I do think that it's both. I think that it can be a way of seeing how much models agree with each other, but also that it is a way of making sure that you haven't done something absolutely wacky with one model that nothing else agrees is reasonable.
So a general principle would be like you'd expect some signal in all of the models, but then just the overall like maximum is.
Yeah.
Thanks.
I'm just curious if there's a biological reason for why, or the model design reason for why that outlier exists with Informer and CTCF. Is it something specific to CTCF binding sites?
I think that,
yeah.
So I think part of it is that CTCF is a very conserved, somewhat simple. I mean, it's not super simple, but it's like it is more simple than some of the other ones in terms of what the motif is and how strongly CTCF binds to all instances of CTCF. As for why Informer and not the other ones have such a strong response, I would say that just is because BPNet and Informer here seem to agree more on the exact composition. Right. I think I have to keep going if I want to get to some of what I think is the most interesting aspect of this. So because of Ledidi's speed, we can use multiple models simultaneously in the design process. Sometimes the way that research goes is that you train a model specifically for the targets that you want to make designs for, even if that's multiple things. Because Ledidi is very fast, you can use multiple models. Maybe you want to use an Informer model, but one of the heads happens to be bad. So you train your own specialized model for that one head. Maybe you have an internal experimental track that you want to train a model for, but you want to combine it with models that your collaborators used, et cetera. And so Ledidi is able to use these multiple models simultaneously to really precisely control multiple characteristics of the design. So the question becomes, which multiple models should we use? Well, I spent the last year in Vienna at the Research Institute of Molecular Pathology, sitting next to Alex Stark. And so given that, I was contractually obligated to use his STARR-seq models. And so we use those in order to design cell type specific enhancers in Drosophila. So here we use two deep STAR models, one of which was from a previous published paper that we didn't touch at all. The OSC model had to be retrained for this work, but was not originally proposed in this work. But each one of them only models a single cell line. This isn't a multitask setting. This is two distinct models. And we wanted to try to design enhancers that were active in only one cell line. But like I said before, one of the aspects of Ledidi is that it is designing edits to an initial template sequence. And so a natural question is, what should those template sequences be? The simplest such set of template sequences is regions that are never enhancers, regions that are non-enhancers in either cell line, and we want to turn them on in one cell line, turn them on in the other cell line. And when we do that, we can see that computationally, we are able to design enhancers that are quite specific. On the top, we have the designs which are S2 specific, and the burgundy shows the predictions for those sequences. The green shows the predictions for OSC, and we can see that those are quite low, even a little negative, and we see a little bit of the reverse. This is in silico, so these are predictions from the model. But we can consider other template pools as well. We can take regions which were initially enhancers in S2, regions which were initially enhancers in OSC, ones that were enhancers in both, and then just uniformly randomly generated DNA. So not endogenous at all, just uniformly randomly sampled. And in each one of those cases, it seems like computationally we were able to design cell type specific enhancers. But the question arose earlier, what about experimental validation for these? So we then went and Franziska in Alex's lab went and ran an actual STARR-seq experiment. And so this is actual real STARR-seq data. The dots correspond to the target cell line that we are trying to design activity in. And what you can see is that the margin between the two is quite large. For the OSC activity, the ones in green, we can see that the activity in OSCs is quite large and the activity in S2s is quite restrained. The ones in burgundy are a little bit more of a complex story. Ultimately, we use this loss, which was trying to maximize the gap between the two, as opposed to necessarily keeping the off-target cell line low. And so that's why we get the results that we see here, where some of them quite a few seem to have activity in OSC as well, but stronger activity in S2. But it's just like across all of these different settings, we were able to design cell type enhancers. We then considered a more complex setting, what we're calling an affinity catalog, where not only are we trying to control the cell type specificity of the region, we're also trying to control the dosage in the on-target cell line. And so we set specific targets here where we want to design weak enhancers, mid-strength enhancers, strong enhancers, and unnaturally strong regions, which are more strong than any endogenous enhancer from previously characterized STARR-seq experiments. We also wanted to see if we could design enhancers which are active in both cell lines from each one of these pools. And so this was kind of a comprehensive experiment. One of the benefits of STARR-seq is that it's super high throughput. And so we're able to easily do thousands of such candidate sequences. This is what the data looks like there. It seems much more cell type specific here because we are explicitly trying to keep the off-target cell line off. But we see a gradient of activity in the on-target cell line, where there's a bit of overlap there between the weak S2 enhancers and the weak OSE enhancers. Some of them kind of dropped out, but that otherwise it's forming this sort of L shape. When we consider the enhancers which are supposed to be active in both, they kind of follow the diagonal, where the regions that they're getting stronger and stronger in both cell lines. So what was kind of surprising here is how few edits we were able to do this in. These sequences were 249 base pairs long because that's the input window of the deep STAR models, but that we were able to design cell type specific enhancers that were of strong activity in fewer than 20 edits. So by editing less than 10% of the sequence, we could induce, kind of strong activity. The min gap ones are a little bit of a outlier there because they are, I can talk about that later, but basically they are a little bit of an outlier due to the way that the objective is formulated. But it was quite surprising how few edits one was able to use here. But this brings up a natural question of, well, okay, so what are the edits that are being made here? And so I want to share two stories here that I thought were really interesting. The first was when we took a region which was initially an S2 enhancer, and we wanted to edit it to become an enhancer in OSE. You can imagine that this involves two components. The first is we have to turn off activity in S2, and then we have to turn on activity in OSE. And so when we look at the attributions from the deep STAR models, you can see that the reason that these models think that this region is S2 specific initially is because of the presence of these two GATA motifs. GATA is a S2 specific factor here. When we want to design enhancers which have weak OSE specific activity, you can see a few things are going on. The first is that we make an edit on the left-hand side to knock out that GATA binding site. We make two edits on the right-hand side to knock out the second GATA binding site. And then we make a few edits that the model is a little bit confused as to what exactly is going on there, but we'll see in a second. With 10 total edits, we were able to convert this from being a strong S2 specific enhancer to a weak OSE specific enhancer, where the experimental data verifies that. I'm not showing the experimental data, but it verifies that. When we want to create a mid-strength OSE enhancer, we can see we're basically doing the same thing where we're knocking out the GATA binding sites on either side, but with one targeted edit in the middle, we have induced a traffic jam motif. Now, it's called traffic jam because you know how Drosophila people can be sometimes. So what's interesting here is that with one targeted edit, just adding in that C, we were able to get a mid-strength OSE enhancer here. But it took us 2 edits before in order to get a weak OSE enhancer. And the reason is because that C site there was critical for getting the traffic jam motif at all. And then you had to make a subsequent edit to make it a weak affinity version of that motif. So sometimes activity in the genome when you're doing editing isn't necessarily linear with expectations. So it's not exactly linear with the number of edits needed for the amount of activity that you want. It takes 10 total edits to get to either direction, and then 17 total edits to make it a very strong version of this OSE specific enhancer. Basically what we end up doing is adding in a second copy of this traffic jam motif, sort of on the dead body of the GATA motif that we had on the left-hand side there. So then the second one that I wanted to share with you was where we took a region which was initially an OSE specific enhancer, and we wanted to make it active in both cell lines. And so this involves adding in activity as opposed to swapping which cell line is active in. Here, you can probably figure out which one of the figures is being included in the paper and which one was made just for the slideshow. But here we can see the attributions for the S2 cell line in the bottom and for the OSE cell line on the top. The reason that this was an OSE specific enhancer is because of the presence of the traffic jam motif with a little bit of other stuff going on the right-hand side there, right next to it. When we want to try making this weak in both cell lines, we can see that not much is really going on. There's some stuff going on off screen here. When we want to make this a mid-strength enhancer in both cell lines is when we start to see something really interesting. By making these edits, adding in a C and an A, we create a traffic jam dimer. This is a very strong OSC specific signal there. But by positioning the two traffic jam motifs at that distance, what you end up doing is implicitly creating an AP1 motif within them. AP1 is specific to S2 as well. And so this is something which I'm describing as basically like Marioska motifs. That these are motifs that are explicitly within each other. By being cell type specific for different cell lines, you can now make them active in multiple very distinct cell lines. This only happens because Ledidi is trying to minimize the number of edits that are being made. You could imagine that if there was no constraint, you could just put the AP1 motif wherever. You wouldn't have to think about this. But it saw that by making these two edits, you could add in activity in both these cell lines at the same time. So I think this is really interesting. Sometimes we think about motifs as just like, oh, there's a blah motif here and there's a blah motif over here. But I've been trying to push for a while now that the genome is much more continuous than that. So here we have totally different motifs that are overlapping each other. Different proteins are binding to the exact same nucleotides in different cell lines. And then we start to see stronger activity in both when we want these to be strong, and then we want them to be unnaturally strong. We see another version of the same phenomena with the TJ dimer and the AP1 motif on the left-hand side. So this is a kind of a reproducible sort of phenomena. All right. I'm going to just skim over a few things, just some food for thought for people. One of the other in silico experiments that we did is that we paired together a Chrome BPNet model, which made predictions for accessibility, with a BPNet model that made predictions for the binding of an individual protein. And by doing this, we're able to design regions that were accessible and control why they were accessible, instead of just using a panel of different motifs depending on what happened to be available. We were able to create sites which were uniformly accessible, but control which exact transcription factors were binding and causing that access, or were binding in those accessible sites. We don't have experimental data for this yet, but these in silico data looks quite nice. We can also turn this concept on its head. We've been talking so far about using models that make predictions for different sorts of things and being able to control different parts of biology with them. But what happens if you use multiple models that make predictions for the same thing? This can be used for a more robust design setting. That if you make edits that all of the models have to agree are helpful for a particular task, like the binding of a particular protein or accessibility, you're much less likely to overfit to the eccentricities of any specific model. This is a little bit difficult to interpret. We're reworking the figure, but the gist is basically that when you use even one other model, when you pair together two models that make predictions for the same things, you get designs which are much more robust and much better validated by external models than if you use any individual model. Okay. This last part is a little bit complicated, so I think that I'm just going to wrap it up at this point. And I think I'm just going to wrap it up at this point and thank all of my and summarize it. So basically, Ledidi is a design method, which means that it can be paired with any machine learning model or a collection of machine learning models to design edits to an initial template sequence that can control very precisely the characteristics of that sequence. We have introduced this idea of an affinity catalog, where instead of just designing for a single target, which is pretty common right now, trying to design very strongly cell type specific activity, you try to make designs for many different steps along a path, because this can be quite useful not only in practice, but also for understanding the biology of sequences. So I think that in general, the development of these sorts of models, these sorts of methods which make use of the trained models are important for the field. We've been spending a lot of time trying to optimize the predictive performance of the models that we train. We have competitions that try to, encourage development in this, but we don't really know what to do with these models. Maybe not don't know what to do, but much less focus has been paid to what exactly we do with these models after they have been trained. And so design, I think, is a really promising area for how we can use these models to actually make a real world impact. So I'd like to thank all my collaborators over the years that have helped with this project and many other collaborations. And thank you for taking the time to listen to me. Thank you.
Thank you for the great talk. I think this is related to Or's question a little bit, actually. A lot of these sequence to function models are trained on the reference genome, right?
Yeah.
And in the past, the reference genome, the reference allele didn't necessarily need to be the most frequent one observed in the population, because I don't think we've had sequenced that many. I read that the newest, like the most recent version, updated some of the reference alleles to the more frequent alleles. Anyway, so I think some of the models are maybe trained, may have been, I don't actually know, trained on an older reference genome. Some of them may be trained on a newer reference genome. And I'm assuming your initial sequence is the reference genome. How much can you comment on whether the consistency between the models with respect to the base sequence used in training matters?
Yeah.
Yeah.
This is a great question. Something you may also be interested in is that I think that Otto Butt had a paper a few years ago showing that the HG19 reference genome had the markers of type 1 diabetes in it. And so for anyone who is studying diabetes using that reference genome, it may have given an incorrect impression of the genetic landscape there, which is weird because the way he phrased it, at least, and I don't know if this is true, it just seemed like nobody had looked before to see whether or not these sorts of disease markers were present in reference genomes. As to whether or not this affects machine learning models, I think the field is kind of split. The intuition makes sense, as you clearly presented, that if you train on the reference genome, but the reference genome is not correct, how could the machine learning models be correct? But when people go and they try to train on either corrected versions or personalized versions or using a variety of genome, it doesn't actually seem to increase predictive performance that much. So I think it's still an open question of whether or not we haven't yet found the best ways of improving predictive performance given the variation that we're able to incorporate, whether or not we haven't actually gotten that much data that really incorporates personal variance, or if there's just something else that we're missing. Because it is true conceptually that any limitations in the data will become present in the models, but this is also, it's unclear whether or not if you're averaging over tens of thousands of protein binding sites, whether or not the variation at any particular or like even a dozen sites really matters that much.
Thanks for the great talk. So I guess my question is related to whether you've done experimental validation on other models which have more nonlinearities added. So I guess for Ledidi, the benefit is you have a pre-trained model and you're basically optimizing a given sequence using that. So and it's speedy. You can plug in different models as well. But have you compared performance on experimental data with, maybe you have a generator network, for example, and then you have your pre-trained predictor.
Yeah.
And have you compared that and you see any improvements or? Yeah, I'm just curious if you run those comparisons.
Yeah, I think the short answer is that no, we don't have experimental data for these sorts of generative models. There are some practical reasons. The first being I don't have a wet lab. But there are other, just intuitively, I am sometimes skeptical about this idea of these generative models because what you have to do is train a whole separate model for your design task against the Oracle model that Ledidi is using. And so it's basically like stacking spinning plates on top of each other. I already don't trust the machine learning model that I'm using Ledidi on. And now this idea of having to train and quality control a whole second model before I can even do design, even if it was successful, it seems to me that it would be a huge investment of time that isn't necessary because ultimately you can just use a method like Ledidi in order to get designs as well.
This is probably a bit of a silly question, but you talked about how Ledidi is a design algorithm and you can pair it with any machine learning. I was wondering how exactly do you plug this machine learning model? Like how is it for the user to do that, for example? What kind of integration does it actually entail?
Or to talk about the implementation details, that if you have the model as a PyTorch object, you just drop it and the initial sequence into the Ledidi function, as well as what you want the output from the model to be, and you're done. So all you need is the PyTorch object. If you want to use multiple models simultaneously, you basically just can create a wrapper class. We have one provided for you where you just put in a list of models, and then what the corresponding output would be. You end up like concatenating or whatever. It's not always straightforward, because if you want to have one model that makes predictions for a profile and another model that makes predictions for a single scalar, you can't just concatenate those, but you can do it with Ledidi if you follow the guidelines.
Thank you. And the second question, on this, on the final part of your presentation, where you talked about combining models like Chrome BPNet and BPNet, for example, and combining where the chromatin is accessible and why it is accessible. I'm guessing what you find is a subset of the two, a small one, I'm guessing, or I don't know how small. You have open chromatin, but only for a subset of that you can actually figure out why something, if actually something is binding there and why. I was wondering if you have or thinking about any examples where you can do some biological validation of why this is interesting, or if there is any way of pursuing this.
Well, in terms of why it's interesting, that most of what this boils down to is the design of constructs for, say, AAVs or other sorts of probes. Basically, that if you want to design something which, say, glows, you want to design something which expresses GFP in proportion to the concentration of a particular protein, then you want, a particular transcription factor. Then you want that transcription factor to bind, and through its binding, increase the expression of GFP. And so if you are designing a promoter site, you want to make sure that there are not secretly, maybe not secretly, but that there just aren't other sorts of binding sites there, so that the signal is convoluted by the binding of other proteins. And so this very specific sort of targeting of a TF has quite a few practical applications when you want to design these sorts of reporters.
So there is a lot of as you said, designing this is for in this whole cell experiment or in vivo experiments. So there is also regulation, a lot of regulation inside. It's a lot of things affect each other, right?
Yeah.
So since we are only one step down, I think you have showed okay, we can have a promoter far end of the protein and far out of sequence, and to which the promoter can bind, and therefore increase the activity. But there are multiple steps, cascades of steps under down this initial thing. So how, what could be what could be done to sort of get to that type of simulate?
Yeah, so that's a great question. Basically, the gist is that predicting transcription factor, binding, accessibility, transcription initiation, that's nice, but that's not the whole story when it comes to gene expression. And if we want to very specifically target gene expression patterns, we need to do more than just these sorts of models. So the nice thing about Ledidi is that you could, any sort of model that's differentiable and takes in sequence context, you could use. And so if we were able to model how changing protein binding patterns influence accessibility, and then stack on top of that a model that looks at how changes in that predicted gene expression might change global patterns, you could stack all that together and use that with Ledidi, as long as it becomes a single differentiable unit. The gist is basically that there's a weakness on the predictive modeling side that Ledidi itself can't solve, but when one of us does solve it, you can immediately plug in Ledidi to do designs.
Thanks. I wanted to follow up on the question of practical applications. I think this is perhaps more of a suggestion than a question, but the reporter design is obviously really, really helpful. But in real world applications, there are times when we do want to create different enhancers in engineered cells. The big one is CAR T cells. So Alex Marson and Luke Gilbert, I think, just had this really nice paper where they did joint genetic to make the CAR T and then epigenetic to reduce T cell exhaustion. But of course, the epigenetic, pro of it is that it's not very durable, but a con is that it's not very durable. So in that case, editing an endogenous enhancer in a targeted way to make it more active to activate a given gene expression program is very desirable. And so I think my question / suggestion is, how many places in the human non-coding genome, in human enhancers, could you predict that maybe 1 or 2 base changes, because that's probably what's practical, right, is possible to create, to go from a weak enhancer to a strong enhancer, or to toggle an enhancer based on your minimum, maximum effects? Because that would be super interesting to know.
That's a really good question. That's something we haven't quite looked at yet because we weren't sure what to do with the results once we had them. It's nice to publish a list of such edits, but it would presumably require experimental validation in order to reach its full impact, which I don't yet have a collaborator in that space. It's a really good idea, though.
So that was really interesting. Thank you. You mostly talked about recruiting activatory factors to enhance expression. Have you tried to design repressor elements?
That's a good question. We have not tried to design repressor elements yet, in part because it's challenging to know whether or not you've designed a repressor or you just haven't designed an activator. And so that would require careful design where you have something which is slightly active, and you need to make sure that you also haven't encountered dropout just of the signal, right? This is something I talked with Alex about when we were trying to design the experiments, and ultimately we decided that because of all these caveats when it comes to repression, that it may be more challenging than an initial paper would, but it's super interesting. Cool. Well, if there aren't any other questions, I'll just mention that because I'm a new faculty member, I'm always looking for new grad students and postdocs. So if anyone's interested or knows someone who might be interested, happy to chat.